Polytene chromosome in a chironomous salivary gland. Polytene chromosome. Polytene chromosomes were first reported by E.G. Balbiani in 1881. Polytene chromosomes were found in dipteran flights and best understood are those of Drosophila, Chironomus and Rhynchoceara. They are present in another group of arthropods of the class Columbola, a protozoan group Celiophora, mammalian Trochoplast, and antipodal and suspensor cells in plants. In insects, they are commonly found in salivary glands when the cells are not dividing. They are produced when repeated rounds of DNA replication without cell division forms a giant chromosome. Thus, polytene chromosome form when multiple rounds of replication produce many sister chromatids which stay fused together. Polytene chromosomes of interphase are seen to have distinct thick and thin bending pattern. These patterns are originally used to help map chromosome, identify small chromosome mutation, and in taxonomic identification. They are now used to study the function of genes in transcription. In addition to increasing the volume of the cell's nuclei and causing cell expansion, polytene chromosome may also have a metabolic advantage as multiple copies of genes permit a high level of gene expression. In Drosophila melanogaster, for example, the chromosome of the larval salivary gland undergo many rounds of endo-reduplication to produce large quantity of adhesive mucoprotein before pupation. Another example within the fly itself is the tandem duplication of various polytene bands located near the centromere of the X chromosome, which results in the bar phenotype of kidney-shaped eye. The interbands are involved in the interaction with the active chromatin proteins, nucleosome remodeling, and origin recognition complexes. Their primary function are to act as binding sites for RNA pol 2 to initiate replication and to start nucleosome remodeling of the short fragments of DNA. In insect, polytene chromosomes are commonly found in salivary glands. They are also referred to as salivary gland chromosome. The larger size of the chromosome is due to presence of many longitudinal strands called chromonomata, hence the name polytene. Polytene means many-stranded. They are about 0.5 mm in length and 20 micrometer in diameter. The chromosomal strands are formed after repeated division of the chromosome in the absence of cytoplasmic division. This type of division is called endomitosis. The polytene chromosome contains two types of bands, dark bands and interbands. The dark bands are darkly stranded and the interbands are lightly stranded with nuclear stains. The dark bands contain more DNA and less RNA. The interbands contain more DNA and less DNA. The amount of DNA in interbands ranges from 0.R to 25%. The bands of polytene chromosome become enlarged at certain time to form swelling called puffs. The formation of puff is called puffing. In the regions of puffs, the chromonomata uncoil and open out to form many loops. The puffing is caused by the uncoiling of individual chromomere in a band. The puff indicates the site of active gene where mRNA synthesis takes place. The chromonomata of puff gives out a series of many loops laterally. As these loops appear as rings, they are called Balbiani rings after the name of the researcher who discovered them. They are formed of DNA, RNA, and a few protein. As they are the site of transcription mechanisms such as RNA polymerase and ribonucleoprotein are present. In protozoa, there is no transcription since the puff consists of only of DNA. Occurrence. The polytene chromosome are present in secretory tissues of dipteran insects such as the Malpighian tubules of Sierra and also in protist plant mammals or in cells from other insects. Some of the largest polytene chromosomes described far occur in larval salivary gland of the chironoid genus Axarus. In plants, they are found only a few species and are restricted to ovary and immature seed tissue such as in Phaseolus oceanus and P. vulgaris, and the another tapetum of Vigna.